Hello, good evening, students. So I hope you all are well. And today we are gonna to discuss about the secondary growth in the dicot stems. Okay? Secondary growth in plants. So let me give you brief about secondary growth first. You have gone through the primary growth. Okay, as the primary growth it occurs in all the plants going through the primary growth, but when it comes to the secondary growth only occurs in the certain plants, in the certain plant group of the plants. Especially secondary growth can be seen in the dicot plants and gymnosperms. Okay, in angiosperms it occurs in dicot plants only and gymnosperms. Especially this secondary growth, it occurs in those plants which have the presence of the cambium tissue. Okay, so cambium tissue is important. Cambium tissue is important for the secondary growth. Okay, so any plants where the cambium tissue is going to be formed, in those plants secondary growth can occur. So these plants, they are open for secondary growth when they have this cambium tissue. Okay, so remember, this cambium tissue, it can occur, only occurs in which plants? It occurs in dicot plants, dicots and gymnosperms. Okay, so those plants, then they are open for the secondary growth. They are open for secondary growth. Okay, so remember, because they have a presence of cambium tissue, when they have the ability to form the cambium tissue, then they are open for the secondary growth. Okay, remember this point. Now remember, the secondary growth, when it occurs in the plants, this secondary growth occurs in the lateral form, lateral stage in the plants, in later, not in the primary growth. When primary growth is what I told you, primary growth is the initial growth of the plant. When the plant is going through the initial growth, all these structures are green in the plants. Okay, there is not a much wood formation, there is not a girth formation in the plants. Okay, but gradually when the plant starts to mature, then the plant's girth is going to be increased. Okay, its diameter is going to be increased. This increase in diameter occurs inside the plants then only when they are going to form this cambium tissue. Now I told you in the beginning that there are the main three tissues are there who are responsible for the whole growth and development inside the plants. Okay, there are meristematic tissues, there are the permanent tissues. Okay, then I said there is also the cambium tissue. There is also the cambium tissue. So cambium tissue, by this cambium tissue, there in the meristem, I told you there are different forms of meristem, different types of meristem. Okay, there is a primary meristem, there is a secondary meristem, there is a lateral meristem. Okay, so when plant at the during the maturation time, there occurs inside a lateral meristem, lateral meristematic tissue, lateral meristem. So later on this meristem gets developed inside the plant. By this lateral meristem, it lead the formation of cambium tissue. It led the formation of cambium tissue. So cambium tissue formation is depending upon the synthesis of the lateral meristem inside the plant. And then by this cambium tissue inside the plants, plant develops the secondary tissues. Plant develops the secondary tissues. Now plant entered into the secondary growth. So those secondary tissues will lead the secondary growth inside the plant. That secondary growth increases the diameter of the plants. Diameter of plant goes increased. So plant increases the girth. The girth of the plant is going to be increased. ठीक है ना? So that is how the secondary growth occurs inside the plants. ठीक है? Now, if we talk about dicot stems, then inside the dicot stem, how this cambium tissue is going to be formed, which is mainly responsible for the secondary growth. How it is going to be formed in dicot stems as well as roots also, because cambium is also present in root as well as stem also, and for both gymnosperms and dicot. So let's talk about how this cambium tissue, how all over the secondary growth occurs within the dicot stems. Okay, I hope you might have understood the main concept, what is the role of secondary growth inside the plant. Now we will go more deep and let's try to understand different structures and functions, how it occurs during the secondary growth in dicot stems. Okay, now first step, the secondary growth in dicot stem, it gets completed by different steps. First step is formation of cambium tissue. So the first, how this cambium is going to be formed, that's the first step to understand how the secondary growth is going to be accomplished. Now second is formation and activity of cambial ring. ठीक है. Now second step, once the cambium tissue is going to be formed, then this cambium is going to be formed in the form of a ring. When the cambium becomes mature, then it becomes in the form of ring manner. So that's called cambial ring. So how that ring is going forward, and after that what is the activity and function of that ring, cambial ring, to let the formation of secondary growth. That is the second step. And third step is formation and activity of cork cambium. Now this is the third step in the secondary growth, that there is another type of the cambium. One cambium is called vascular cambium, which is going to be formed in the ring manner. This is vascular cambium. There is another form of the cambium going later on developed during the secondary growth only, and that is going to be formed to the in the peripheral tissues of the plant. Okay, that is known as cork cambium. Okay, how that cork cambium is going to be formed and what is the activity of that, that is the another step. In these three steps, the whole secondary growth is going to be accomplished inside the plants. Okay, so let's understand stepwise. First we'll understand how this cambium is going to be formed inside the dicot stem, formation of cambium tissue. Okay, so let me rub this side. Till then we complete this first step, then I can move ahead to the second step. Now, formation of this cambium tissue inside the dicot stems. Now you have understood. If I will give you the overview of different layers which you have gone through already, you have gone through the anatomy of root, you have gone through anatomy of stem, you have gone through anatomy of leaf as well. Okay, now you know the different zonation which occurs inside the root, stems and leaves. Okay, so if I take, this is the outermost zone, outermost layer of the plant. Fine. So consider this outermost layer as epidermis. This is epidermis. Fine. Then after the epidermis, there is the cortex region. Okay, so you consider that this much area. Fine. Consider this much area is okay. This is the cortex. This is the cortex layer. Fine. So just make it in dotted form. This is the cortex. Okay. Then later you have gone through, after the cortex, inside there is the endodermis. Fine. Consider this layer as a endodermis. This is endodermis. Fine. Now inside the endodermis, now all these tissues inside the endodermis, what is this region called? I have already told you, this whole region, it is known as stellar region. It is known as stellar region. Fine. So inside the stellar region, in this stellar region, there is another layer which is going to be formed. Fine. The outermost layer of the stellar region, here, this is called pericycle. Okay, which I have already told you. So you know that all these zonation are inside present in the stems, dicot stem. Now in this stellar region, there is the arrangement of the vascular tissue. Okay, and you know that vascular tissue in dicot stem, it is going to be formed by vascular bundles. And the arrangement of vascular bundle in the dicot stem, all the vascular bundles are arranged in a ring manner. Okay, so like this. So this is the arrangement of vascular bundle in a ring manner. Okay. Now also I have told you already, these vascular bundles in the dicot stem, the vascular bundle, these are conjoint type. These are conjointed. And conjointed means what? Conjointed, I told you, conjointed vascular bundles have the presence of xylem tissue and phloem tissue within the same bundle. Like this. So you know always the xylem, this central region here, this central region which is vacant over here, this central is known as pith. As you know that this is a central pith. Around this central pith, all the vascular bundles are arranged in the ring manner, and inside each vascular bundle there is the presence of xylem tissue which is nearby towards the central pith. Like this. Xylem always gets formed in these vascular bundles nearby the central pith in the vascular bundles. Like this. So this is the xylem tissue. Fine. And in each, you know that this is the primary xylem. If we go particularly which xylem it is during the primary growth, it is the primary xylem. Okay. Now, as of now, secondary growth has not yet started. So the xylem is primary xylem. Okay. Around this primary xylem, within the same vascular bundle, there would be the phloem tissue outside of the xylem, like this. In each bundle, the arrangement of xylem is endork system. As you go gone through, what is the endork system? Protoxylem lies towards the center, metaxylem lies towards the periphery, that is protoxylem. So xylem is always inside within the vascular bundle and phloem is towards the periphery. This is the phloem tissue. So again, this is primary phloem. Primary phloem. You know that this is how the vascular bundles are arranged inside the plant during the primary growth. During the primary growth. Now, when the secondary growth starts within the plant, then what happens then? Within these vascular bundles, each vascular bundle where there is a xylem and there is a phloem tissue. So in between the xylem and phloem tissue within the vascular bundle, some of the cells become meristematic here and led the formation of a meristematic strip-like structures between the xylem and phloem tissue. Between the xylem and phloem, there is going to be formed a meristematic tissue in the strip manner, in the small strip manner like this. Between the xylem and primary phloem, in between that, like this. And initially this meristematic tissue becomes in the formed in the form of strips. This strip form, patch form of meristematic tissue which is going to be formed, this is a cambium tissue which is going to be formed within the xylem and between xylem and phloem tissue within the each vascular bundle. And this cambium, it is known as intrafascicular cambium. It is called intrafascicular cambial strip. Okay? Remember this point, intrafascicular. It is also called fascicular cambium, but to be very specific, particular, which is going to be formed here between the xylem and phloem but within the vascular bundle, so it is called intrafascicular cambium. Okay, now first, within each vascular bundle, the intrafascicular cambium is going to be formed in the patch manner. Okay, now look here, this area between the vascular bundles,